perform negative control. From what we call metastatic gate, site scattering versus CD45, as you can see here. This is a typical dot blot in alveolar rhabdomyosarcoma, metastatic carcinoma, and plasma cell myeloma. As you can see, this is the granulocytes, lymphocytes, monocytes, and also this is the blast gate. All this area, the R4, is the metastatic gate. And as you can see, we have huge numbers. So we are not talking about few cells per ml. We're talking about many cells per ml. Countable, measurable, and definitely we can quantify them. That's all about the minimum residual disease test. Concerning the research activity, RGCC works as a company of contract research organization. So we can measure substances as candidate drugs wannabe. So we evaluate, as I said before, in specific cell lines and perform cytotoxic assays or viability assays fast, so that we know if a drug is a substance is a potential drug or not. Also, we perform the majority, of course, of the assays, since they are simple. And we have also in-house a storage facility for many types of cancer human cell lines. So we can pick many types of our wish, perform the test, and we know which way we proceed further on animal test or more accurate test. So this can become very attractive, or it can become more attractive. And this is few of the assays that we work already. THP12, it's tetrahydropyranose 12, is a carrier for cisplatinum. It comes from a university in Germany. It's a new formulation of cisplatin to make it more lipophilic, trying to make it more effective for prostate cancer, because this is a problem in prostate cancer. Another product, it's Anvesal, is a product from a company called Nerium Incorporated in the United States. Is an extract from oleander, and it works very well in prostate cancer cell lines, which we are very close to perform a study for many types of cancer as well. And here we have a compound come from a British company called CV247. It's a combination of simple substances that it seems that it has a natively cytotoxic effect as single agent, very effectively. And the most impressive thing is that when it's used with an already established cytotoxic drug, it makes it equally effective in very low dose. A very desirable feature from pharma industry and for a candidate drug. So I'll pick a few examples. Since I'm in Britain, I pick the example from the British compound. Yep. Here is the decrease rate, is the killing rate, as it's known. Here we see the cisplatin in 1 micromolar, 5, 10, and 50. 50 micromolar is high concentration, lethal. It's not used in clinical practice. But as we see here, a dilution 1 to 100, when we combine it with cisplatinum, we may achieve 100% decrease rate equally to the lethal dose, which means that you may have 1/5 decrease of the concentration of cisplatinum and sustain the benefit. And as we know, cisplatinum is a very good drug, but with very nasty toxicities. Nephrotoxicities, ototoxicities, neuropathies, etc. And all of those toxicities are dose dependent. So if we manage to decrease it, but keep the toxic effect in cancer cells, we may have something extremely good. And we already know by experience, by clinical experience, that platinum compounds already search for a cell protective agent. Maybe CV247 is one of those. and basic research, which is cancer stem cells. As I told you, we collaborate with CellProgen upon cancer stem cell-like lines to determine specific features which they become targets. siRNA, very popular. It's a double-stranded small RNA molecule who actually can block and silence a specific target and see if this target is druggable, can lead to cell death of cancer or not. And of course, we involve with delivery systems of those, how we can deliver effectively and selectively these substances to cancer cells and not to anywhere else. And of course, the latest is the transposable genetic elements and cancer resistance. We try to figure it out how this manipulation and rearrangement of genes, how it happens, if this is an outcome of luck or it's a program process which leads to resistant phenotype, which can create a background for further studies and development of new therapeutic approaches. This is the references. I hope that since you already signed out for the attendance, I may have the opportunity to send you by email or by mailing all these references and all these presentations to you as well. Thank you for hearing me. So, I'm all yours about questions. I know that you are tired. It's taken more than an hour. Sorry about that.
How much does it cost to have blood test?
How much does it cost?